Let's begin the project presentations. First up, we have 3D Metabolism. The project group includes Mohsen Movahidi, Joshua Yun, Ryan Saeed, Kim Nip, and Ella Gray. Before we hear from the student team, I'll first pass the floor over to 3D Metabolism's PI, Dr. Lindsay Rogers. Is she here? Okay.
I'm going to just start our slides. I think Ella and I are going to Can I advance with this too? Yeah. Okay, thanks. Yeah, so I'm gonna just spend the first minute talking about this project, and then I'll pass on to Ella and Mohsen. But yeah, essentially what we're working on is an emerging media-based project to more effectively visualize cellular metabolism and support learning in that space. So for some of you that aren't biologists here, we define metabolism as kind of the entire collection of chemical reactions that functions within a really dynamic, complex network within sort of every cell in every organism to enable kind of every biological process. So it forms this really complex network, and it's kind of applied across a lot of fields of biology. So students learn metabolism first in high school, and then in biology and biochemistry and pharmaceutical sciences during post-secondary education, and also in sort of research settings and medicine. So we thought there was this huge opportunity to learn to use emerging media to really enhance and improve kind of how we can visualize and represent metabolism. So if we look over here at sort of the existing learning tools that existed before we began this project, we have sort of a couple options. So this is sort of a diagram of a metabolic pathway from a textbook, so a 2D image. And this is what we refer to as a metabolic map. So here we have kind of a visualization of the network, and here we have sort of just one pathway sort of isolated from this network. And really what we're trying to do is kind of combine the information, sort of the detail that exists here, as well as sort of the network representation that exists here, and also add some sort of exciting additional kind of features and functions that kind of bring this really exciting network to life. And we're doing that using emerging media. So we have sort of 4 project goals. The first is to recreate metabolic pathways in a 3D space. We're also working to kind of compile and sort external data from a wiki base that we've generated, and then call that data to our network. And we're also adding a lot of UI features and functions that enable the user to manage complexity within this really complex network, and also enable the user to visualize metabolic flux. So when I say metabolic flux, I mean sort of the flow of metabolites and reactions kind of as they flow through these pathways within our network. So that's a really brief introduction from me. And I'm going to pass off to Ella Gray next to discuss sort of project goals 1 and 2 here.
Okay, thank you for the introduction, Dr. Rogers. I don't actually know how to go to the next one. There we go, okay. So I am a subject matter consultant, and my job is to take pathways that are traditionally represented in 2 dimensions and convert them into a comprehensive and coherent 3D network. In our network, nodes represent metabolites, so that's like glucose and acetyl-CoA, and edges represent metabolic reactions. Edges connect to nodes to show how metabolites are converted through metabolic reactions, and in each cellular compartment, a metabolite is only shown once, and this allows us to show how a single metabolite can be involved in multiple different pathways and how interconnected metabolism really is. Okay, so if you look on the right of this slide, you'll see a traditional representation of glycolysis taken from a well-known biochemistry textbook, and next to it is our representation of glycolysis in Unity. Here you can see how we've represented metabolism in 3D space and how nodes and edges connect to form reactions that make up metabolic pathways. One of the most exciting things about our project is that it allows the user to appreciate the interconnectedness of metabolism. So you may notice that there's some extra nodes and edges surrounding glycolysis in Unity that aren't highlighted, and they're not part of the glycolysis pathway in the traditional textbook representation. These nodes and edges belong to other metabolic pathways that work in conjunction with glycolysis. So that means they might produce metabolites that glycolysis uses, or they might use metabolites that are produced by glycolysis. If you were limited to learning biochemistry from a textbook, you'd have to flip through pages in order to learn about these other metabolic pathways. But with 3D metabolism, it's all right there, and it's easy to appreciate how connected they are to glycolysis. Okay, so you might notice that there's some chemical detail that seems to be lacking from Unity that's seen in the traditional textbook figure. This information is found in the Wikibase. The Wikibase is an open source software best known for being the software behind Wikipedia. It can be accessed by the user to find all of the information that's not included in the Unity model. Each metabolic pathway has its own Wikibase page that contains a description of the pathway and all of the reactions involved. And clicking on a reaction opens up a new page that contains tons of supplemental information about that reaction. And together, the Wikibase and the Unity model contain all of the information included in a traditional textbook, but represent it in a much more connected and coherent way. Okay, I will pass on to Mosa now.
Thank you so much, Ella. All right, so I'm gonna talk a little about the tech stuff that's going on in 3D metabolism. I'm the software developer on the team. But before I start, I just want to acknowledge the work that our UI UX designer has done, Kim Nip, and also the work that our old software developer, who's now a supervisor over at the Cedar Lab, Dante Cerrone, has done. He's in the back. Hi, Dante. Yeah, without those two, like, none of this work would be possible. So thank you so much. But yeah, as my two colleagues mentioned, this is a very complex network that we're trying to replicate on this tech side of things. And we kind of need to let the user or researcher student to focus on a specific part just because how complex this is. And this is why we have the highlighting feature and the label feature. So as you can see in this small video, the user can choose the pathways they want to focus on. They can even choose only to focus on the specific one, so they can toggle the labels. They can say, okay, we have two modes of highlighting. One of them is yellow, one of them is blue. And the yellow version actually shows the directions of the reactions as well. So if they want to hyper focus on a few pathways, but they want one specific one to see how that relates to these other ones, they have the ability to do so. On the next slide, however, I want to talk to you guys about one of the integral part of our project, which is animations. And the reason we have animations in this project is we want to kind of visualize the flux through these reactions in a way that like you can't really see on a textbook. On a textbook, you can't really play a video, as you all know. And we kind of built this animation editor onto the Unity software, Unity being the engine that kind of runs all of this tech. Which is called IET4U, which basically is this custom editor that any user can use to grab simple animations that we pre-make and kind of put it in a sequence. So for example, here, you can see that they're kind of like getting highlighted. There's a little wiggle, and it kind of shows the flux through this, which is called the mallet aspartic shuttle as part of the pathways. All of these are like tiny animations here, like stop wiggling, start wiggling, which is very simple for the user to kind of understand. But then once we sequence it, it creates this whole thing, which shows the reaction going through. But we kind of wanted to take this to the next step. And that required us to change our database. And as Ella mentioned, we have kind of 3 types of data types. So we have nodes, which are metabolites, edges, which is our reactions, and pathways, which are kind of like the groupings of these nodes and reactions together. And we had to create this hybrid graph. And for those who are not necessarily in computer sciences, graph is essentially a way to replicate network on a data sense, on a data backend. But this didn't really fit into a traditional set of definition for a graph. So we had to like actually consult with the Faculty of Computer Sciences, and we figured out a way to connect these. So the reactions hold connections for the metabolites in these data types. And I don't want to bore you guys with the details, but the nodes don't really hold any information other than the information about themselves. and pathways are the ones that connect the metabolites to the reactions. And this is how like the computer algorithm goes through. And this gives us the feature of custom animations. And custom animations is something that we're currently working on, where we actually, instead of us pre-making animations for the user to see, so if you press the button play and then it plays it for you, we actually let the user choose what animation to see. So they can choose any two metabolites on our network. So for example, they can choose this glucose up here and something down here, and the computer will automatically create the animation for them. So this is kind of like we give the customizability to the user. But yeah, I'm gonna pass it back to Dr. Rogers to send us on.
Okay, I'm just gonna finish. Thanks, Mohsen. So I'll just finish up with a few sort of mention of future direction. So we've been working on this project for almost exactly 2 years. The technical ability at EML is really amazing, and I think it's amazing how far this has really come in that time. But we are still working really hard on this project. So we have some directions that we're currently going. So importantly, we're still working on expanding our network. We sort of initially focused around carbohydrate or sugar metabolism, sort of as a proof of concept sort of place to start. We'd really like to expand the network to include additional metabolic pathways relating to sort of lipid or amino acid metabolism. Importantly, because it's going to be really exciting when we get this tool to the point where we can really effectively represent physiological states. So disease conditions such as diabetes or particular cancers, once we can really model and represent that using this tool, I think it's going to be sort of a huge step for us. We're also working to incorporate sound into the network, designing a website to host the tool, designing a lot of learning tools and other learning resources and other kind of instructional resources to support the use of the tool, and also sort of collecting some data as we implement the tool into classrooms. We're working on sort of a web-based application now, but eventually we'd like to expand into either an AR or VR version as well. And I guess we really like the idea of potentially using a similar approach to also visualize additional networks. So potentially neuronal networks or networks in computer science, or even sort of networks sort of in root systems and soils. So just to finish, I just want to acknowledge all the people that have supported this project, particularly at UBC Studios, Saeed, Danikar, Kim Nip, and Michael Sider. We've had a lot of support from Emerging Media Lab, particularly from Dante Siran, Catherine Winters, Daniel Lindenberger, and the students on our team that have been sort of actively involved in the project this term are, where's Joshua? Joshua Yoon, Ryan Saeed, Ella Gray, and Mohsen Movahedi. Oh, and I also want to thank the UBC Teaching and Learning Enhancement Fund for funding this project.